Hey everyone, welcome to another episode of Darshan Talks. I'm your host, Darshan Kulkarni. It's my mission to help patients trust the products they depend on. So, if I'm an attorney, I'm a pharmacist, and I advise companies with FDA regulated products. So, if you have an interest in a company with FDA regulated products or the products themselves, this is the podcast for you. Today's podcast is going to be about the history of antibiotics. And it's one of those interesting topics that came up because we've had Doug Bucklin on our podcast before, and Doug has been really interesting talking to us about intellectual property, about patents. And I was watching his LinkedIn feed and noticed that he posts a lot about the Federalist Papers. He had a whole historic bend to it. I said, why don't we try something different? Why don't we actually have a discussion about history? Why don't we actually see, especially in this time of COVID, and I recognize that antibiotics don't work with viruses. I'm a pharmacist, believe me, I get that part. But it's just an interesting conversation to have on antibiotics, the history of them, where they came from. Is there a future in antivirals? Do we sort of mix those two things together? So if you are a CEO of a company that is developing antibiotics, if you are just a history buff, if you will, this is the conversation for you. Our guest today, I mentioned, is going to be Doug Bucklin. He is a senior attorney at Volpe Koenig. He actually works in the life sciences, helps with a variety of different technological areas, everything from pharmaceuticals, biotech, new chemical entities, apparently carbon sequestration, food technology, nutraceuticals. Essentially what Doug said to me, and I really like the way he framed this. He's a science-minded attorney who brings science from the lab into the hands of people who need it. And I think that's a really great way to look at how do you bring value to individual patients. Because despite what everyone thinks, that's what drives most of us in the life sciences. We really just want to help people. That's why we got in here. It does. I mean, I still have to make a living. I still have to do all of those things. But it's nice. I mean, the reason we do it is to help people. So, ladies and gentlemen, our guest for today, Doug Bucklin. Welcome, Doug.
Thank you, Darshan. It's nice to be here. And I'd like to echo, this is one of the few industries where people enter to be do-gooders more than at a higher frequency than others. Life science is a very optimistic industry, and I really enjoy being in it because of that.
Yeah. It's one of those weird things where we and I have these conversations on trust, and the life sciences industry does exceptionally badly at it. We're not a very trusted industry, even though our goal is to help patients. And I never quite understand how those two work out, but they do. And it doesn't help that sometimes data shows that we don't fudge our numbers properly. I can never tell if it's a question of Monday morning quarterbacking or and someone going back in and going, well, you didn't do this right. And yeah, you know what? They probably didn't do it right. But how many things would be fined as wrong if we went in and double checked everyone's calculations again? But in this case, I think that there is a good reason to look over our shoulders because we're in the business of saving lives, and that's important.
Right.
So anyways,
let's not go down that rabbit hole right now.
Absolutely. So now talk to us a little bit. Let's start off with antibiotics. What are they for everyone that doesn't know? And more importantly, we all know what antibiotics are, but what are not antibiotics? And what I mean, that's a weird reverse question to ask, but we all know of the average mom or dad who walks into the doctor's office and says, little Timmy is sniffling, give him an antibiotic, and that's what I'm here for. And if you don't give it to me, you're practicing medicine badly. Is the antibiotic even right for that? What are antibiotics is the crux of the question.
Broadly speaking, antibiotic means something that's anti-life. So it could be an agent that is targeted against bacteria or fungi, protozoans, or people. I mean, broadly speaking. So when little Timmy's going into that doctor's office and has a cold and the parents want an antibiotic, they're usually off base nowadays because the antibiotics that we use and we're familiar with are antibacterial agents, and viruses are what cause colds. So an antibiotic will not help little Timmy's cold. It might help if he's getting a secondary infection because of a bacterium, but it's probably best not to just indiscriminately prescribe antibiotics. And I think that makes a nice segue to broadly why are antibiotics important. So
Yeah.
Think of this statistic. For every single man, woman, and child on the planet, each one of them has 50 tons of bacteria that could be allocated to them and them alone. That's how much more bacteria live on this earth than you, me, and everybody else. Now, to bring it to the personal, each human being has roughly 10 to the 10th cells. You carry with you roughly 10 to the 11th bacterial cells. So there are more bacteria on you than there are cells of you. And each of those bacteria is actively growing. And as it's actively growing, each time it replicates its DNA. Each time its DNA replicates, it makes errors. Lots of those errors, the vast majority of those errors are corrected. But given the number of bacteria you have and the errors that are made, there's roughly 300 to 1,000 errors in new progeny bacteria on you. So when you take an antibiotic, if one of those errors happen to be in the target of that antibiotic, the organism harboring that mutation might just escape killing by that antibiotic. And if it does, it will grow, flourish, and become the dominant population, and you'll have antibiotic resistance. So antibiotics are wonderful. To rewind on that story a bit, it used to be that if you had a bacterial infection, you'd likely die. It wasn't uncommon that people would receive a scratch. It would get infected and they would die. Very tragic history. So I don't want to lecture too much, but if you want to jump in and ask a question, that's great.
Yeah.
But
I was sort of letting you do your thing because there's so much there to explore. I guess the big question I started thinking about, and maybe this for a thin man like you, that's not a big issue. But for me, as I keep gaining weight after COVID-20, after the COVID-20, I keep going, well, what does that actually mean? So what I'm really thinking about is you talked about we've got more bacteria than cells on us. There are 50 tons of bacteria for every person out there. Does that mean that most of my weights are attributed to the bacteria that I don't even see and I try not to feed?
Well, you can try convincing people that, but no.
So let's frame the question differently. One of the big areas that you really get into in the area of bacteria is in the area of GI bleeds and stuff. And one of the things you talk about is the use of vancomycin, the indiscriminate use of vancomycin, and how that's causing bacterial resistance. And the idea is that you now have to use things like probiotics to counteract the effects of antibiotics. So maybe we want to talk a little bit about where did we start. What have we always understood as bacteria? How did we start with this is the problem? Like, how did we realize that bacteria were the problem to start off with? And then how do we start sort of going after them? And we'll kind of go from there.
That's a long history. The germ theory of microbiology.
Yeah.
Go back to Louis Pasteur and experiments done. People used to think that like flies and maggots came spontaneously generating from rotten meat, old meat. But then experiments were done to show that no, they didn't. That if the meat was isolated, that the maggots would never develop. And zip ahead, Pasteur and Cook and other microbiologists came up with the germ theory. And then, so nice thing to remember is Cook's postulates. Cook was a scientist who found that if you're going to say something causes a disease, you should first be able to take from the organism afflicted and isolate what you think is the causative agent, grow it separately, and then put it into another organism and see that the other organism comes down with the disease. And so from Pasteur and Cook, we have this idea firmly rooted that microorganisms cause disease. And so then how do we get to antibiotics? Well, antibiotic history kind of parallels. Antibiotics didn't just fall out of the sky. The news way back 2500 BC, there's evidence that they would use mold to treat infections. And that mold, we know now, had an antibiotic in it. Egyptians were said to have beer that had antibiotics and mold in it. I've got another one here I wanted to bring up because it's just so rather disgusting. Where is it here? Babylonian doctors used to heal eyes using a mixture of frog bile and sour milk.
Well, the question is, did that work though? They used to heal it, or at least they tried to, but how many people really just let it blindness?
Well, I think from the writings, there's some idea that it did have some effectiveness. So we have to look back in history and say, does it make sense? And the idea of rubbing mold into a wound is really kind of disgusting to us right now, and there's no way you'd do it. But it seems to have worked. And Russians used to, poor peasant Russians used to warm up soil and rub it in their wounds in order to treat them. And in hindsight, that might have introduced actinomycetes and other fungi that produce antibiotics. And so it might have helped. So zip ahead, there was a guy
I'm not
named...
sure. I mean, I know a lot of Russian people, and I love my Russian friends, but they are a hardy bunch. And I'm kind of going, that's just, you had an arm chopped off, rub some salt, some mud into it, you'll be fine, just keep going. It reminds me of that Monty Python sketch. It's just a flesh wound.
Little kids always said rub dirt in it,
Yeah.
That's kind of hit or miss. You might have ended up putting something that would kill you in your wound, but
yeah.
So around the turn of the 20th century, a guy named Paul Ehrlich in Germany noted that he could dye bacteria different colors. So some dyes would absorb into some cells, but not to others. And he had this idea that you could develop a compound then that selectively killed bacteria. And so that was kind of the opening on this rational search for a compound that will kill bacteria selectively. And he came up with arsenic. There's around the turn of the 19th to 20th century.
Got it. Okay.
And so his arsenate compounds became like the way you would treat syphilis. And not terribly nice compounds, but it was kind of the opening salvo. And again, parallel to that, people had noticed that, microbiologists noted that if you had a particular fungi growing on a plate, it seemed to inhibit bacterial growth. So then coming up to like the bottleneck point in antibiotic research was the development of penicillin. And so Alexander Fleming, who's a British scientist, he's always described as a guy that was a lazy microbiologist who left things lying around and went on vacation and came back and found that there was something interesting on one of his Petri plates.
Before we get to Fleming, let me ask you a question,
Mhm.
because this sort of is interesting to me. The idea of arsenic to act as an antibiotic. As we mentioned early on, you talked about arsenic. Sorry, you talked about antibiotics as being anything anti-life, the bio referring to life, and it's very reminiscent of current cancer treatments. We use arsenic to treat cancer. It's anti-life. In some senses, what you're almost saying is many methodologies in current cancer treatments are literally antibiotics.
But of chemotherapy is you develop a compound that selectively hits the problem, whether the problem is a bacteria or a cancer cell or whatever it is. I mean,
Right.
your chemotherapy is meant to develop, in this sense, a small molecule that goes and binds to a target and does something interesting to it.
Which
yeah,
is,
I mean,
it's also reminiscent of some of the conversations we had during COVID, where people said, "Why don't we use bleach to get rid of the vaccine?" I mean, one of my favorite ads. I just saw this. I forget, one of our very, very famous cleaners, like the actual.
I lost sound. No, it's not.
Can you hear me now?
Yes.
Okay, thank you. One of the really interesting things that's happening right now is during COVID, we talked about the use of material of cleaners, and there's a very famous cleaning substance. I can't remember which one, but they came out and said cleans 99% of bacteria, including and viruses, including COVID-19, and it's because it's a type of bleach, and it just cleans all the surfaces completely. So is there a difference between, like, can we just call everything that kills something an antibiotic, or is that just too indiscriminate a word? Should we be more targeted in the terminology to actually count it as an antibiotic? Does that make sense?
I, we can get into cutting hairs and deciding how to divide things. I mean, as it's used today, the word antibiotic means a drug that is going to kill an invading organism. And usually that invading organism is a bacteria. The bleach, there's bacteriostatic and bactericidal solutions. Bacteriostatic slow the growth of bacteria. Bactericidal kill bacteria. So bleach is something that kills bacteria. Now it will also kill us. So it's not a very good drug to consider taking. So the bleach is great for disinfecting surfaces, and maybe even if you need to on your hands, if you rinse your hands afterwards, because it's also caustic. But it doesn't make a good drug. So for chemotherapeutic uses, you want something that's going to target the microorganism, but not the person. And I think that was part of what Paul Ehrlich was getting at way back when he noticed dyes preferentially staining different cells. That here's the fundamental idea is that we can make molecules that target one thing and not another. And those arsenate compounds targeted bacterial preferentially. It wasn't just arsenic, it was an organic compound that included arsenic as a moiety. So it was specific for bacterial cells.
So as we now, you're starting to talk about Fleming. I want to pull us back, get into Alexander Fleming. How did we go from a world of bactericidal or just in general cidal products like arsenic to maybe we can be a little bit more focused? And I think Fleming sort of originated that in some senses.
But I think Fleming was he was at the cusp of the most successful story. So he went on vacation. He came back and he saw this petri plate that had Penicillium notatum growing on it. It's a fungi, and the fungi, where it was growing, there was no bacteria growing. On the edges where there was no mold, there was bacteria. So he thought that was interesting and he tried to trace it down. Other people had noticed similar things, but he actually was able to do some rudimentary purification techniques and come up with something that, even if he diluted it 800-fold, would kill bacteria. And so Penicillium notatum was recognized as something that produces an antibacterial agent. So then he published on it, he developed it, and other people started picking up on it. Now that was in 1928, but everybody knows that antibiotics were not widely circulated until World War II. So why did it stall out?
Yeah.
The root problem there is chemical engineering. The amount of penicillin that Penicillium notatum produces is just too small. So let me look at my notes over here to get the names here because they're important people. Oh, I had them highlighted here so I could tell you. Ernest Chain, Sir Howard Florey, and another fellow named Boris. They picked up where Fleming left off, trying to improve the situation, and they got to a point where they thought they could treat a person with penicillin. And so they found this person. His name was Albert Alexander. He was a police constable. He got scratched by rose thorns in the face. Some people doubt that that's actually how he got scratched, but he got scratched, and he had an infection that was closing off his eye, and it was threatening his, because if you had an infection from a scratch back then, it was likely you would die. So they had penicillin that they had worked up, and they treated him. And after the first treatment, he got better. He was doing well. The tragedy of that story is that they ran out, and they ran out before they had killed enough of the bacteria. So the bacteria took over again, and Albert died. So it was an encouraging step towards using penicillin, but it ended in tragedy because they couldn't produce stuff. So this great potential of using penicillin as a magic bullet to cure bacterial infections wasn't coming to fruition because it was technically not possible. So, but it was an interesting thing, and people around the world were working on it. And so there's this.
Let's take a step there, though. I did an experiment. Someone died. This is the first time I've ever tried this. What makes me go, I'm going to try it again, especially in 1928?
Well, he did get better when they had administered the agent.
Okay.
The problem was that they didn't have enough agent to continue the treatment.
Okay. You just didn't have enough lazy people like Alexander Fleming, is what you're saying. Growing more. We didn't have enough lazy people like Alexander Fleming, is what you're saying. Right.
Well,
which is mind-blowing to me that he was lazy. That was awesome.
It comes up in the history. I bet he was a fabulous researcher, and people just use that story, and it's repeated over time. He won a Nobel Prize. I mean, the guy wasn't a slouch. But a lady has come to be known as Moldy Mary. What was her actual name? Looking over at my notes again. Mary Hunt. She worked at the U.S. Department of Agriculture's Northern Regional Laboratory of Research in Peoria, Illinois. She had found some mold growing on a cantaloupe in a market. And that mold on the cantaloupe was Penicillium chrysogenum. And the Penicillium chrysogenum produced penicillin at levels many-fold greater than penicillium notatum. So she forwarded the strains to people that were working on it, and this actually was happening during World War II. So soldiers were dying because they would get wounds, they'd get infected, and the infection would kill them. And everybody recognized that, and everybody knew about this research. Not everybody, but people knew about this research on penicillin and other antibiotics, and were trying to make them work. So Mary's mold got forwarded on to the important people. And the U.S. government actually engaged lots of different companies and people in order to find a way to produce penicillin in mass.
But now let's ask a really basic question, because we're running out of time a little bit, so I want to keep us a little tight. One of the big problems we face now is not enough antibiotics. What you described was the birth of an industry, if you will, to cure people.
Right.
What's different between now and then? Why was there an incentive to produce new, better antibiotics then, but we don't have that now?
But before we get there, just let me say that the interesting point of that story ends in Westchester, Pennsylvania.
Oh.
With
by the way, we're both out of Pennsylvania, which, in case that's not obvious to people. So yeah.
So the fellow's name was G. Raymond Retu. He had a mushroom research facility because we grow mushrooms here in southeastern Pennsylvania. It's, I think, the world's leading producer of white button mushrooms still.
Did not know that.
And Retu was interested in improving that process. Well, that process of being how people learned how to grow the mold that produces penicillin in mass in order to make enough of it. And he ended up teaming with Wyeth and made Wyeth, and he served as the leader of Wyeth afterwards for a long time. So the story of penicillin is the story of southeastern Pennsylvania pharmaceutical industry.
Did not know that. Huh.
But Pfizer was involved in it and other groups too. So your question, we had a shortage then, but we wanted to get a bunch. Well, we got a bunch. So if you look at the history of antibiotics, there was a lot of research. We produced a bunch. They were approved up through the 1960s. And then it just kind of fell silent. And there was some work in the 1990s, and it fell silent again. There have been major news events of antibiotics given regulatory approval for a long time. Maybe in the last year or two, I'm not sure. I have to check on that. But I think we slowed down because we were so successful. We now have, but you mentioned this vancomycin. We have vancomycin on the market, and vancomycin is our backstop. When penicillin doesn't work because the organism is resistant, so you have methicillin-resistant Staph aureus. You go to the hospital, you get a drip of vancomycin, and you're saved. You don't die from septicemia.
Not anymore.
Right. And if it's not vancomycin, it's this one or that one or the other. We've got lots of different antibiotics. We feel safe.
Yeah.
But the problem with that feeling is what we started out talking about. That inherently, Jurassic Park, life will find a way. No matter what defenses you put up, because just evolution, life will find a way around those barriers. And eventually, each one of the antibiotics we use will fall to antibiotic resistance. It's just the way it's going to work. It's not a if question, it's a when. Eventually, penicillin won't work anymore for most of the organisms that are a problem for us, because resistance will be so widely spread, and there'll be different resistant mechanisms. So we'll need something else, and then something else, and then something else. I think that's why there's renewed interest now in antibiotics. And you started out talking about COVID, and I think that's a great way to tie it all back. As people sat in the hospital with COVID, which leads to secondary infections, which can be a huge problem, antibiotics were just in droves. And chances are that we developed some new resistant strains or enriched populations of resistant strains by doing that. So COVID may be leading to yet other problems with other pathogens.
So what you're saying is, though, that we were the victim of our own success.
I think so.
But now we know that we have depleted our options. We need new options. But companies are saying things like, you know what, antibiotic treatments are 14 days and 21 days or even less than that. And there's just not enough of a market out there. And
it's hard to make a blockbuster drug on it.
Correct.
It's not a chronic treatment.
It's not a chronic treatment. I'd much rather be investing into one pill once a day for the rest of your life, like a statin, for example. That's the general mentality. People point to COVID and they say, you know what, we had data that was developed based on public money, and drug companies came through. We got some vaccines out of it, but drug companies already talking about raising the cost. Why are we not able to? And the FDA is trying to take certain steps, for example, regulatorily, where they're saying that we'll provide certain regulatory incentives for investing into antibiotics and bringing antibiotics to market. But let's be honest, none of these are actually super successful. No one's coming up with. Go ahead.
Have you seen the Pasteur Act?
Please.
This was introduced in the last Congress. I don't know if it's been reduced yet. But the point of the Pasteur Act is to incentivize new antibiotics. So, company files an IND. If it's, and then they file also a request to be covered under the Pasteur Act. If that's granted, then there's a guarantee that their subscriptions are going to be worth, I think it was $750 million to $3 billion. So there's a known remuneration at the end of it. And how they're going to recover that money, I think there's a hot button political issue there that's going to hold up the bill, because it's going to feed into people who are on federal health care plans. It's going to be covered by federal payments. People who are not are going to have to pay for it to cover that cost. That's going to be a political mess. But the step back from that, the incentive to create the antibiotic is they're trying to create it with the Pasteur Act, guaranteeing $750 million to $3 billion in remuneration. But will it work? Will it pass? I don't know.
Politically, do we even have the will? And socially, do we have the will? I mean, politically, yes, we're putting the act together. But I can see people going, you're basically guaranteeing money to pharmaceutical companies who are making billions off of these things. You're guaranteeing billions. Were there better places to put that money?
Well, I think you always have to remind people that pharmaceutical companies, yes, they make a lot of money, but they also spend a lot. To get that drug out on the market, it costs up from $1 billion up, and they're going to have 10 losers. So they have to recover 10 billion when they put one drug on the market. Now, that's maybe not for every case. Maybe it's not all the way up to 10 billion, but it's in the billions to get one drug to market.
Yeah.
They need to make that back, or they need to fire everybody and not produce any more drugs. So it's a choice. Do you like medicine in the state it is right now and want to see no more advances, or do you want to, let the companies develop products? Yes, there are bad pharmaceutical companies, and we have rather obnoxious examples of CEOs extracting extortion payments from people.
Yeah.
But I think by and large, they're good companies, and we need to remind people of the economic interplay of how costly it is to get a drug out there, and that if they're being guaranteed $3 billion on the back end, that's roughly about the same cost you'd be paying anyways. We're just restructuring the incentive system to make the payments at a different point.
So let's sort of we're beyond where we normally should be with our time. What's a question you would want the audience to answer for us?
Audience answer for us. One is I'd like to learn about all the efforts for new antibiotics. So if you're involved in new antibiotics, I want to know about it, because I think that's just technically interesting, and that's where my interest is really peaked, on the molecular side of things. So if you know that, great. But I'd like to know more about the Pasteur Act and other incentive systems that are being talked about in order to make antibiotics an attractive economic venture. We have a company here, Venator RX. They produce new antibiotics. They have drugs in the pipeline. So how are they able to exist and make a go at it? So what models are going to work for small companies?
So during this podcast, we landed up talking a little bit about the history of antibiotics. We talked a little bit about how those antibiotics, literally the concept of what an antibiotic is. We then went into things like what antibiotics became. We talked about cidal versus static, and then we got into some of the political things that are going on right now around incentives and bringing new antibiotics to market. Did I miss anything? That I tried to cover that.
It's pretty good.
Okay. Nothing in this podcast should be construed as legal or clinical advice of any kind. It's for education and entertainment purposes only. For those of you who missed it, Doug, where can they find you?
I'm at Bucklin at vklaw.com. And Volpe Koenig is my firm. If you search that name out, you'll find the website. You can click through the people to find me and find my phone number and everything.
Awesome. And if you like this podcast, please leave a comment. Please subscribe. You can find me on Darshan Talks on Twitter or on our website at DarshanTalks.com. Thanks again for coming on, Doug.
Thank you, Darshan. See you.
See ya.